In order to characterize the color of all the images that we have on file, we need to find the average hue of a certain section of each wing. And in order to do that, I'll just quickly show you how hue is measured. So this is a graph here of hue. It's measured in degrees, so going from 0 degrees up to 360 degrees. Most of our hue values are going to be somewhere in here, obviously. If we have a very, very bright image, it could show up somewhere here. But in general, we're going to see something around this region. Before we do anything, I'll have you guys make an Excel spreadsheet. Probably put 3 worksheets down here, one for Danaus Euripus, one for Danaus Plexippus 1, the first folder, one for the second folder. And you can map them out like this. The ID, that's very important. We keep the IDs together. The mean hue, mean saturation, and mean brightness. Those are the 3 values that we'll gather. So let's say for ID number 1, we're going to go ahead and use Photoshop. So open Photoshop. You would open up the ID number 1 that we have from the Danaus Plexippus folder. Now here's our image. In order to select a cell, and I'm actually going to have you guys use the distal cell of the forewing and not the hind wing. I've decided that I think that this is a better representation of the color for our images. So we're going to go ahead and select the wand tool, and I've found that generally a tolerance of around 50 works pretty well. You might want to, you can mess with it up or down a little bit, but then just select the cell. And you can see that it gets the cell there. We want to make sure that it's not containing any of the black, it's only the orange, and that's fine if there's a speck or two that's not selected. We just want to get the average of this cell. Now in order to find the average, we're going to calculate the average and then fill this selection with that average color. In order to do that, we just need to click Filter and then press Average. And you can see there it kind of takes that image and gives us the average color for that entire selection. Now in order to quantify this, we just go down to the color picker. So we click the foreground color. We use this dropper, make sure that the sample size is on point sample, because obviously we already have the average, so anywhere in here is going to be the same, and we want it to just be a point. And then go ahead and just press down, and you can see that this has an average hue of 20 degrees, saturation is 88%, and brightness is 61%. So then we can go back and we can add those in here. 20 was the degrees, 88 was the saturation, and the mean brightness was 61. We can go ahead and format these cells as percentages just to keep everything straight in the data file. Just put them in as decimals even. That could work as well. So essentially we just need to make sure that these data files are kept tidy so that when we go back and we say we have ID number 1, then we can look that up in the cardinalide database that I have and match that and say this butterfly has a mean hue of 20 degrees and a cardinalide content of whatever it may be, and that's how we'll do our comparative analysis. So if there's any questions, you can let me know. Essentially, I'll open up another one here and do a second example. So if we open it up, and this is the Danaus Plexippus number 1 folder, we just did ID number 1. So let's say we do ID number 2. We want to make sure we're using the dorsal surface and not the ventral. And these samples, most of the ones you'll be able to tell pretty easily. As you can see here, this is a common dorsal. The images are a little bit, or the colors are a little bit bolder. If you look at that same butterfly on the ventral surface, you can see it's pretty pale, especially in the hind wing. And also, there's a V here for ventral. So we don't want to be using that for this particular study. We'll be wanting to use the dorsal, which does not have a marking here for a D, but it's pretty obvious that this is dorsal. So if we open up an ID number 2, we select with our magic wand tool the distal cell of the forewing, making sure there's no black there. Then we go to filter and average. It's going to select the average and fill it back into the selection. Go back to the foreground color picker. Make sure it's on point sample. Select a point within the selection. You see that this hue is a 23 degree. So if we go to ID number 2, 23 degrees, 78% saturation and 63% brightness. One thing that will also help us is if we go ahead and add a column in for gender. I would highly recommend that. So the first one happened to be a male. We can just say M. The second one happened to be a female. We'll just keep it in order that way. And in order to tell if it's a male or a female based on our images, there's a gender symbol at the bottom of each image. Again, if you have any questions, feel free to contact me, and we could even meet if you'd like a in-person tutorial on this process. But I do think that Photoshop is going to work the best for us rather than ImageJ. And it should go pretty smoothly as long as the data is kept tidy.